am a director and founder of the nonprofit foundation, Agricultural Genomics Foundation, which aims to bring cannabis science to a broad public. I am also the co-founder of the Cannabis Genomic Research Initiative, which is our group at CU Boulder, and my research is funded by the company Steep Hill. And today I'm going to be talking about the governmentally produced cannabis. This is my contact information, my email, my Twitter, my website. I want to start by thanking our collaborators at CU, also our collaborators from the industry. And cannabis is a plant that's an angiosperm, so it's a flowering plant. It is dioecious, so males and females are found in different plants, although there's monoecious populations, so hermaphrodites that exist. It is wind pollinated, pollen travels through the wind. It is annual, so it leaves and dies during a year. However, there's populations in tropical and subtropical climates that may live more than a year. And last year we had this article published, which is a review of the medical literature that says that cannabis is useful for 3 different conditions: pain in adults, nausea and vomiting in chemotherapy, and spasticity in multiple sclerosis. So it's missing things like epilepsy or like the sedative effect, so the couch effect, or and these are very, cannabis is very known for treating these other types or producing these types of effects. We also had this article published in the New York Times about a month ago that says that cannabis can be used for cancer, for epilepsy, or for soothing, for example, menstrual cramps. And this is just one of the compounds, so this is just CBD, which is one of the compounds produced by the plant. The plant produces multiple compounds. Some say that it can produce even 100 compounds between terpenoids, which are usually responsible for the smells, and cannabinoids, which interact with our bodies. 2 of the wide-known cannabinoids are CBDA and THCA. The plant produces them in the acidic forms, and when we heat them up, they turn into CBD and THC. These are the forms that interact with our bodies. That's why, you know, we bake it or vape it or smoke it, because we need to heat them up, right? THC is highly regulated governmentally. Legally, hemp must have less than 0.3% THC in the US. So why do we have this discrepancy between the scientific literature and the popular culture? You know, we're accepting 2 different stories about the plant. And it's either because the popular culture is wrong, or it's because the scientific literature, we're studying what we should not be studying. So most scientists must use the cannabis that is produced by the government, by the National Institute on Drug Abuse, NIDA. This is a subdivision of the NIH. And basically this is how it looks like, right? Like, this is the governmentally produced cannabis, and this is the cannabis that we can find in dispensaries where cannabis is legal across the US. In 2017, we published this paper where we compared the cannabinoids produced by the governmentally produced cannabis to that from the private markets. So this is, Steep Hill produces this. This is what they offer to their customers. In this case, this is from this variety called Girl Scout cookies. It has this cannabinoids and this terpenoids. So this is the service that they offer to their customers. And therefore, we were able to compare with data from Steep Hill. We were able to compare the private market to that cannabis produced by the National Institute on Drug Abuse. So we had 4 locations for the private markets. We had Seattle, Sacramento and Oakland, Denver, and that produced in the University of Mississippi for NIDA. So the University of Mississippi has a contract with the government, and they are the only ones that produce the cannabis that we can study at in universities, particularly for medical research. And this is what we found in that paper from 2017. In this first panel, we have CBD. In the second panel, we have THC. NIDA is the second box in orange. We can see that the median It's the lowest one for both CBD and THC. Now, for CBD, it differs from everyone significantly except from Seattle. For THC, it differs from everyone. So it's the one that produces the lowest amount of CBD and the lowest amount of THC. Now, if we look at the whole variation in cannabinoids in this principal component analysis, we can partition this variation into two, the gray group and the pink group, and we can see that NIDA is found in only one of the groups, so only the pink group. And if we partition this variation by location, we can see that Oakland and Sacramento capture NIDA's variation, but NIDA does not capture Oakland and Sacramento's variation. However, NIDA does have a lot of CBN. And CBN is this breakdown product that accumulates when you leave your cannabis under poor conditions, usually under heat. So it's the compound that usually causes the headache and the drowsiness. So NIDA does have more of that one. So we conclude in this paper that NIDA's cannabis lacks variation and potency, contains more of the breakdown product, it is not used for medical or recreational purposes, and has very little phenotypic variation. So therefore, research with NIDA's cannabis is not accurate or representative. Some claim that cannabis can help be part of the solution of the opium epidemic. The head of NIDA, Nora Volkov, says that there is no evidence for such claim, and there's no evidence because NIDA is a Schedule Drug 1 under the DEA. So Schedule 1 drugs are ones that do not have accepted medical use. And it's part of the Schedule 1 along with LSD or ecstasy. And when they're a Schedule 1 drug, they are the ones that have the most potential for abuse. And when you deschedule it from Schedule 1 to Schedule 5, then Schedule 5 has the least potential. And to get a Schedule 1 drug, you need an FDA license, a DEA license, you need a safe, a vault, several types of locks, you need a camera at all times, etc. So it's really hard to get cannabis to study it, even when it comes from the government. And when you get it, as is the case for Sue Sisley from Arizona, who she had to resign because of her cannabis research. So when you get it, it can come with mold after 2 years. So she applied, and then 2 years later she got it and it was moldy. Our collaborators from the University of Northern Colorado got a hold of DNA from 2 different varieties. This is their preprint that you can find in bioarchives. And with 10 microsatellite loci, what they found is that NIDA's varieties, here in blue, cluster with hemp. They conclude in their paper that people consuming NIDA's varieties may experience different effects than the average consumers. We got a hold of that cannabis and we sequenced it, these 2 varieties from NIDA, and this is what we found in this graph where we see the relationship between many different varieties. We can see that NIDA's varieties here in green are very close together. There's very little variation compared to everything else, so we cannot study all of this, we can just study this. And they're very different from everything else. So cannabis has a lot of phenotypic variation. It can be used in multiple settings: fiber for clothing or paper, biofuel, beauty products, of course, medicine. These are leaves from different varieties, and the number of the leaflets change, the colors, and not only the leaves but the plant themselves are very different, their sizes, their colors, and of course all of the multiple products that the plant produces. However, we cannot study all of that diversity. And this is just from the plant point of view, but from the medical point of view, people are using now very concentrated types of cannabis, right? Dabbing or vaping. And we really do not know about the long-term effects of marijuana use. And for my purposes, the questions that I am interested in is the one species. We really do not know if there's any kind of isolation. The evolution of sex chromosomes, as I told you, there's males, females, and hermaphrodites that can interbreed. Where is cannabis originally from? There was a recent paper that suggests Asia, but we don't really know. Why does it produce all of these compounds? What is the ecological function of all of these compounds? We do not know. So we need to keep on fighting. We need to maybe get our DEA license, our FDA license, and then maybe get our terrible NIDA's weed to study something that is not representative. And we need to keep on fighting because now in Colorado, or at least in Denver, they legalized mushrooms. So I bet that we know even less about these guys than we know about weed. And we need to keep on fighting and elect politicians that accept scientific evidence. And in that positive note, there's some that are fighting for us scientists to be able to do our jobs. And with that, stay tuned for more research. Again, this is my contact information, my email, my Twitter, my website. Please donate to our nonprofit organization. Donations are tax exempt. We have a 501c3 status. And thank you very much.
Can you talk more about your funding and your relationship with your funding sources?
So my funding comes from Steve Hill mostly. Still, some of it comes from CU, but mostly comes from Steve Hill, which is this private company that provides a service to people, right? Like if you have a dispensary, by law, you need to test your cannabis, not only for the compounds, but also for mold or for heavy metals. And they are the ones that fund my research. Yes?
Do you have any plans to look at the transcriptome differences?
We have several transcriptomes and we've been looking at them. That is not a project where I'm involved, but yeah, there's some transcriptomes that we've been looking at, but not from NIDA. I mean, from NIDA, we got this DNA somehow, you know what I mean? So unfortunately, we cannot look at the NIDA transcriptomes. Yes.
So you talked about dispensaries testing their own product. Is that how you got data from different states with the dispensaries' data, or did you?
From Steve Hill. So the dispensary itself cannot test the cannabis. It's a third party. So you take, you take it. In Nevada, for example, it's a testing facility that goes to your dispensary. In Colorado, you take it to the testing facility, and there's multiple. It's a service that they offer, and the different states require different cannabinoids or different terpenoids or different types of molds or heavy metals.
And is that where you got your data on like cannabinoids and THC levels?
Yeah, from Steve Hill. Yes, which is the company that funds my research. Yeah. Thank you.